Cellular responses to external stimuli heavily rely on the set of proteins that are expressed in the cell surface in a given moment. Accordingly, the number, the subcellular localization, and subunit composition of these proteins are dynamically controlled. Here, we will use an antibody-feeding approach to quantify the level of surface-expressed receptors in cell cultures as long as the internalization and recycling ratio. This is a very versatile protocol that provides mechanistic information of the regulation of surface-expressed proteins. In our example, we will study glutamate receptors in primary hippocampal neurons.
This protocol can be adapted to any protein possessing an antigenic extracellular epitope. If antibodies aren't available, transfection of tagged constructs can be useful for both labeling and studying specific mutations. In our examples, we use antibodies against endogenous GluA1 and tagged GluN2B.
To begin this procedure, transfer the cover slips cell side up to a paraffin film-covered tray to save reagents and facilitate manipulation. Save and maintain conditioned media at 37 degrees Celsius for incubation and washing steps. Incubate the cells with primary antibodies diluted in conditioned media at room temperature for 15 minutes. After this, use a vacuum pipette to carefully aspirate off the antibody-containing media and wash the cells 3 times with conditioned media.